In 1897, H.G. Wells published The Invisible Man, a story about a scientist who invents a way to change a body's refractive index to that of air, so that it neither absorbs nor reflects light. For over a century, this remained the stuff of science fiction. In the real world, biological tissue is stubbornly opaque. Skin, muscle, and bone are complex matrices of water, fats, and proteins that scatter light like headlights in a dense fog. But in late 2024, a team of researchers from Stanford University and the University of Texas at Dallas turned fiction into reality. They did not use a futuristic metamaterial or a gene editing tool. They used FD&C yellow number 5, also known as tartrazine, the same orange-yellow dye found in Doritos, Mountain Dew, and gummy bears. By applying a solution of this common food dye to the skin of live mice, they achieved optical clearing, rendering the skin transparent enough to see the beating heart, the peristalsis of the intestines, and the intricate firing of neurons in the brain. When the dye was washed off, the skin returned to its normal opacity with no apparent harm. This is the story of the Tartrazine Effect, the physics that makes it possible, the history of our quest for invisibility, the controversy it has already sparked, and the revolutionary future it promises for medicine. To understand why the Tartrazine Effect is such a monumental breakthrough, we must first understand why we are not transparent. Why can we see through a glass of water or a jellyfish, but not through our own hands? The opacity of biological tissue is not due to light absorption, at least not primarily. If you hold a flashlight to your hand, you see a red glow. Light can get through, it just cannot get through straight. The problem is scattering. Biological tissue is a heterogeneous mixture of materials with different refractive indices. Water, which makes up the cytosol of cells, has a refractive index of roughly 1.33. Lipids, or fats, and proteins, which make up cell membranes and structural scaffolds, have a refractive index of roughly 1.40 to 1.50. When light travels through a medium and hits a boundary between two different refractive indices, it bends, or refracts. In tissue, light encounters billions of these boundaries per centimeter: cell walls, organelles, collagen fibers. Each time it hits an interface between the watery cytoplasm and a fatty membrane, it bends slightly. This chaotic bouncing of photons is called Mie scattering. Imagine driving through a thick fog. The water droplets in the air scatter your headlight beams in all directions. You cannot see the road ahead, not because the light is blocked, but because the information the light carries, the image of the road, is scrambled. Skin is essentially a fog made of cells. For decades, the holy grail of deep tissue imaging has been optical clearing. The goal is simple: make the refractive index of the fluids match the refractive index of the solids. If water could be replaced by a substance with a higher index, light would pass through the tissue without bending, just as a glass rod disappears when dipped in oil of the same refractive index. Scientists have achieved this before, but always with a catch. Methods like Clarity, Cubic, and Disco work beautifully, turning mouse brains into glass-like blocks. However, they usually require removing all the water and replacing it with toxic solvents or hydrogels. This kills the tissue instantly. It works for autopsies, not surgeries. Mechanical stretching can align collagen fibers to reduce scattering, but this is impractical for whole organs. Applying glycerol can temporarily reduce scattering, but it works slowly and often dehydrates tissue to the point of damage. The scientific community was stuck. We could have transparency, or we could have life. We could not have both. Until Dr. Zihao Wu and his colleagues looked at the problem backwards. The breakthrough published in Science was born from a counterintuitive idea rooted in the fundamental equations of optics. Usually, to make a material transparent, you try to remove color. You want something clear, like glass. Adding a bright orange dye seems like the exact opposite of what you should do. However, the team utilized a principle of physics called the Kramers-Kronig relations. These mathematical formulas describe a deep connection between two properties of a material: absorption, meaning how much light the material absorbs, and refraction, meaning how much the material bends light. The relations state that a sharp change in absorption at a specific wavelength will inevitably cause a sharp change in the refractive index at nearby wavelengths. Tartrazine is a dye that absorbs blue and ultraviolet light very strongly, which is why it looks yellow-orange, because it subtracts the blue. The researchers realized that this intense absorption in the blue spectrum forces the refractive index of the solution to curve upwards in the red part of the spectrum. By dissolving high concentrations of tartrazine in water, they created a fluid that blocks blue light and matches the refractive index of lipids for red light. When this dye diffuses into the skin, it replaces the interstitial fluid. Suddenly, the watery parts of the cell have the same refractive index as the fatty membranes, but only for red light. The result is that the scattering stops. The fog lifts. The tissue becomes transparent to red wavelengths. As Dr. Wu noted, for those who understand the fundamental physics behind this, it makes sense, but if you are not familiar with it, it looks like a magic trick. The images that accompanied the science paper were nothing short of iconic. They did not just show data, they showed a superpower. The team started with a simple phantom, slices of raw chicken breast. As they soaked the chicken in the tartrazine solution, the refractive index rose. Within minutes, the opaque pink meat transformed into a clear, amber-colored jelly. They could read text placed behind the muscle tissue. They then moved to live, anesthetized mice. They rubbed the tartrazine gel onto the scalp. The result was that the skin became transparent. Researchers could directly observe the blood rushing through the delicate vessels of the brain using laser speckle contrast imaging. The skull itself remained opaque because calcium scatters light differently, but the skin covering it vanished. Next, they applied it to the abdomen. The abdominal wall faded away. The researchers watched the intestines of the mouse contracting in real time. They could see the liver and the bladder. They observed the rhythmic motions of breathing and heartbeat, all without making a single incision. Crucially, the tartrazine effect is temporary. It takes a few minutes for the dye to diffuse into the skin. The effect lasts as long as the dye is replenished. When the experiment was over, the researchers simply rinsed the mouse with water. The dye washed out, and the skin became opaque again. Any dye that was absorbed into the bloodstream was metabolized and excreted in the urine within 48 hours, turning the urine of the mouse bright yellow but causing no apparent toxicity. This wash-on, wash-off capability is what separates the tartrazine effect from all previous tissue-clearing methods. However, no scientific breakthrough is without scrutiny, and the tartrazine effect is currently the subject of intense, healthy debate in the academic community. Shortly after the science paper made headlines, a preprint appeared on bioRxiv, challenging the safety and efficacy of the method. These critics argued that to achieve the refractive index match, the concentration of tartrazine must be very high, around 0.6 molar. This creates a hypertonic solution that could dehydrate living cells, potentially killing them or stopping their normal function. Critics claimed that true transparency was only achieved because the high salt and dye concentration effectively fixed or damaged the superficial cells, making them act like dead tissue. They argued that live mammalian cells in a culture dish shriveled up when exposed to such high concentrations. Dr. OU and colleagues have maintained that while the solution is hypertonic, the effect on the whole organism is different from isolated cells in a dish. The skin is a robust barrier designed to handle environmental stress. The reversibility, the fact that the skin returns to normal and the mouse survives healthy, suggests that while the cells might be temporarily stressed, they are not destroyed. This debate highlights a critical nuance. Biocompatible is a spectrum. Chemotherapy is toxic, yet it is used medically. If tartrazine causes temporary cellular stress, but allows for a life-saving cancer diagnosis without surgery, is it safe enough? This is the question defining the next phase of research. If the tartrazine effect can be translated to humans, the implications are staggering. We are looking at a paradigm shift from invasive exploration to optical inspection. Consider the end of blind blood draws. We have all experienced the pain of a nurse struggling to find a vein, poking the needle around blindly. A dab of tartrazine gel on the arm could render the skin transparent. The nurse would see the vein as clearly as a wire in a glass tube. No more guesswork. Consider early skin cancer detection. Melanomas often grow deep into the skin before they are noticeable on the surface. Current diagnosis involves a biopsy, cutting a chunk of skin out. With this technology, a dermatologist could apply the transparent gel to a suspicious mole. Using a microscope, they could look through the upper layers of skin to see the tumor's roots, its vascular structure, and its depth, all without a scalpel. This is an optical biopsy. There is also a laser therapy revolution. Lasers are used to remove tattoos, treat port wine stains, and kill cancer cells. However, skin scatters the laser beam, diffusing its energy and limiting how deep it can go. By making the skin transparent, the laser beam stays focused. It can reach deeper targets with less power, reducing the risk of burning the surface skin, while effectively destroying a deep tumor or ink deposit. We can also imagine digestive and cardiac monitoring, where a doctor can check for an intestinal blockage or an abdominal hernia simply by applying a patch and looking. While deep organs like the heart are likely too far beneath thick muscle for this topical method to reveal fully, surface organs and the abdominal cavity could become accessible to visual inspection. Even wearable health monitors could be transformed. The Apple Watch currently uses green light to estimate heart rate through the skin. If the skin under the watch were transparent, the sensor could optically track blood glucose, oxygenation, and even specific biomarkers in the blood with laboratory-grade precision. Before you buy a gallon of food dye and try this at home, please do not. There are significant barriers to scaling this from mice to men. First is the thickness problem. Mouse skin is incredibly thin, only a few cell layers deep. Human skin is 10 times thicker. Getting the dye to diffuse deep enough to create transparency without taking hours is difficult. Researchers are investigating microneedle patches, tiny painless needles that punch microscopic holes in the outer skin layer, to deliver the dye deeper and faster. Second is the color problem. Tartrazine makes tissue transparent to red light. To the human eye, the treated area looks dark orange or red, not glass clear. You cannot just look through it with your naked eye and see natural colors. This technology will likely be paired with digital imaging. Cameras that are sensitive to red and near-infrared light will capture the image, color-correct it, and display it on a screen for the doctor. You will not look through the patient, you will look at a screen showing the inside of the patient. Finally, there is the safety profile. As noted in the scientific debate, the high concentration of dye required is a concern. While tartrazine is FDA approved for food, rubbing a super concentrated sludge of it on your skin is different than eating a few Doritos. Human trials will need to prove that this does not cause severe irritation or allergic reactions. The Tartrazine Effect is the latest chapter in a long history of humanity trying to see the unseen. In 1895, Wilhelm Röntgen discovered X-rays, allowing us to see bone inside flesh but with harmful radiation. In the 1950s through the 80s, we learned to use ultrasound and MRI to map soft tissue, which was safe but had low resolution. In the 2010s, techniques like clarity allowed us to make dead brains transparent, revolutionizing neuroscience, but requiring the subject to be deceased. The Tartrazine Effect marks the beginning of the live transparency era, combining the resolution of optical microscopy with the non-invasiveness of ultrasound. We often think of biology as wet and physics as dry. The Tartrazine Effect is a triumph of interdisciplinary thinking, using the dry equations of the Kramers-Kronig relations to solve the wet, messy problem of biological scattering. While we are not yet at the point where we can put on an invisible suit, we have crossed a threshold. We have proven that the opacity of the living body is not an immutable law of nature, but a solvable physics problem. As we look toward 2025 and beyond, the line between the outside and the inside of the body is blurring. With a little yellow dye and a lot of physics, we are finally learning to see clearly.